Let us discuss about Fc epsilon R1 receptor. It is the high affinity IgE receptor. It is expressed on mast cells and basophils. It binds IgE with a high affinity Kd 1 to 2 into 10 to the power minus 9 molar. Due to high affinity of Fc epsilon R1 receptor for IgE, it binds to IgE despite the low serum concentration of IgE, that is 1 into 10 to the power minus 7. On a human basophil, between 40,000 and 90,000 Fc epsilon R1 receptors are present. The Fc epsilon R1 receptor contains 4 polypeptide chains like 1 alpha chain and 1 beta chain and 2 identical gamma chains. These 2 identical gamma chains are linked by a disulfide bond. These 2 identical gamma chains form gamma homodimer. The external region of alpha chain contains 2 domains called as Ig-like domains. These 2 domains contains 90 amino acids. These 2 domains are homologous with the immunoglobulin fold structure. So, Fc epsilon R1 receptor belongs to the immunoglobulin superfamily. Fc epsilon R1 receptor interacts with the CH3, CH3 and CH4, CH4 domains of the IgE molecule via the 2 Ig-like domains of the alpha chain. The beta chain spans the plasma membrane 4 times. It is thought that beta chain links the alpha chain to the gamma homodimer. The disulfide linked 2 gamma chains extend into the cytoplasm. Each gamma chain has a cytosolic domain. Cytosolic domain of each gamma chain has a conjuged sequence. The conjuged sequence is known as ITAM. ITAM stands for immunoreceptor tyrosine-based activation motif. The ITAM motif on Fc epsilon R1 receptor interacts with protein tyrosine kinases to transduce an activating signal to the cell. Fc epsilon R1 receptor has a role in anaphylaxis. Now we will discuss about Fc epsilon R2 receptor or CD23 receptor. It is the low affinity IgE receptor. It has a soluble CD23 domain. It is specific for the CH3 domain of IgE. It has a lower affinity for IgE. Kd is equal to 1 into 10 to the power minus 6 molar than Fc epsilon R1 receptor. It has proteolytic cleavage domain. The Fc epsilon R2 receptor plays a variety of roles in regulating the intensity of the IgE response. Allergen cross-linkage of IgE bound to Fc epsilon R2 receptor activates B cells, alveolar macrophages and eosinophils. Please like, subscribe and share.